you the floor.
Yeah, thank you for the introduction, and thank you also for giving me the opportunity to present our small piece of research that contributed cryo FIB imaging, especially on this place, in this place where the most of the recent cryo imaging development happened. So cryo imaging is a technique for imaging of non-contrasted sample, and contrast is based on the charge position on membrane. So the membranes charge more than the surroundings, and the yield of secondary electrons depends on this accumulated charge. That's why we see contrast, and that's why we see also a lot of artifacts on most of the cryo images. And in comparison with the stain sample, the contrast is lower, especially for protein. And benefits. So the main benefits is the simplification of the preparation method. So especially for me, as I'm not good in chemistry, so you just take your sample, freeze it, and go directly to FIB SEM. And of course, the sample stays in near native state. So it is perfect method for combination with cryotomo. And also fluorescent preservation, so you don't have any problems with the fluorescence in cryo. So this is also a big benefit. So this is the image of, probably most of you know the cryo image like this with lots of artifacts. That's why we put a lot of effort to optimize it. So we optimize imaging parameters, geometry, workflow, also software for keep the high quality imaging through the whole volume. So this is the result. The level of artifacts goes down, and we are able also to do cryo imaging in a bigger volumes. So this is the one slide of the collected volume. So you can see all the ultrastructure. Our model organism is the insulin cell. So you can see insulin granules. Also, this Golgi body on the upper left is especially beautiful. And although actin filaments are visible, mitochondria, so nuclear pores are very well defined. So in following slides, I will show you some sample times that has been tested for cryo imaging. So this is another cell. So we freeze it like cell suspension. So we reconstructed almost entire volume of COS7 cell, resulted in 1,000 slices. And you can see very rough segmentation of membrane, and also the mitochondria's lipid droplets that are very well, a lot of charging in cryo contrast. They overcharge, and also the nucleus. So let's go to tissue. So this is the muscle tissue. Muscle tissue is especially nice in cryo. So you can see here and there, okay, whatever. There are Z disc visible at the end of the sarcomere. That was one. And the H band, if I remember correctly, H band in the middle of sarcomeres. And these mitochondria, there was applied some simple post-processing, so you can see some small artifacts around the mitochondria. And again, very rough segmentation. Yeah, the green discs are the discs on the end of sarcomeres. There is a brain tissue. So we have some volume also from brain. Brain is particularly challenging because of a lot of myelin cause overcharging. So in some extent, it is possible to suppress it by post-processing. There is also recent research from local group. I think there is a poster about it, about the interleaved sparse scanning that really helps also to mitigate the charging. And the last slide is about there is an O side, so you can see also beautiful biology here. These multi-physical bodies or whatever, some mitochondria, reconstructed microvillas. We have also some limited data sets from plant tissue, so it's a tomato root, and also hyphae, lift up with hyphae. So how we get there? What are these optimization steps? So first of all, you need to have proper system. So in these experiments, helios hydra has been used. Standard helios with gallium also works, but it seems that immersion mode is important. Immersion mode allows low energy imaging, and also it increased the SE collection efficiency like to almost 100%. I am not salesman of Thermo Fisher, but we really tried hard to acquire this level of quality of cryo images on other systems of other vendors, but we was not successful. So we use low KV imaging and to suppress charging and to improve contrast. The 90 degrees imaging is used in all experiments. So that's why we have to swing the stage on each slice during volume collection. Yeah, oxygen milling seems to have the best performance in terms of curtaining, and also to ensure the high quality images through the entire volume. We the software calculates resolution on each slice, on each acquired slice, and if the resolution drops below defined limit, it invokes some auto functions. So here you can see that how with the increasing imaging angle, the contrast also increases. Of course, with increases energy, contrast goes up as well, but you have to be aware of charging. So that's why we use low kV, 1 kV or even bit below, and 90 degrees imaging. Also, decrease of working distance helps, but it is very complicated to change working distance during serial FIB SEM volume acquisition. So that's why the acquisition workflow has been established. It consists of multiple steps. So the main step is the milling trench creation that allows milling at 90 degrees in respect with the sample phase, and also imaging phase creation that allows perpendicular imaging. This is the image of a trench. And if you really need to reconstruct entire cell, it is very beneficial to have a fluorescent module for navigation. Okay. Yeah, this is the milling rate. So there is some comparison of milling rates of different species. So xenon is fastest because of high atomic number. Gallium also works for low current, but for high current, it is really slow probably because of energy spread because of spot shape and spot size. And it is important to know that these results are valid for our geometry. Like milling rates heavily depends on the angles, also the state of your precursor. So that was valid for the geometry we use. We also tried to calculate the curtaining score. So you can see why we use oxygen, because oxygen produce low curtains even in higher currents. But like I would say that all species are usable. Also gallium on lower currents works well. So of course depends on the use case. Similar research was also done here we have a little bit different score for the calculation of score. So it is basically standard deviation of filtered image where are only curtains present. So curtains are this vertical line, like artifacts from milling. Yeah, you probably know that. So last thing, what I would like to show you is this software development. So it turns out that to maintain like really high resolution images through the long multiple days volume acquisition, it is quite difficult to do it with the standard tools like AutoSlice and View. So that's why we came with this. It has some features. Surprisingly, it's very helpful also for standard resin room temperature samples. So the main workflow is like this. So you acquire first number of slices, like 10, 20 slices by standard acquisition scheme with some auto functions in defined region and I don't know, drift correction by temper matching, so very standard things. Then you segment your region of interest in your favorite segmentation tool, mine is MIB, and you train your model, deep learning model or whatever model, export it, and Maestro, yeah, it's called FIB SEM Maestro because it is Maestro in cryoimaging, and Maestro will use it for segmentation on each acquired slice. And it is very helpful because if you want, if it is needed to calculate the resolution, it is more precise to calculate it just in your region of interest. It is also faster. Also, the auto functions happen only in your region of interest. Drift correction, I have some cool animations about it. Also, this animation says nothing, but the drift correction. So it basically center your segmented region into the center of a field of view. So it acts like feature tracking, and also field of view optimization. It really speed up the process because it optimizes field of view based on your region of interest. So you just acquire only your region of interest, so it can speed up the whole process. Yes, and this is particularly good for cryo, this auto function, because so the standard auto function works on series of images in some defined area, and this is not very not perfect for cryo because of high dose, and you probably would barbecue the area. So that's we implemented this feature. So you basically change the working distance or any other variable. So you sweep the variable during scanning, during one scan, and then the resolution of each line is calculated and the best line is selected. So by this you can do auto functions in one scan. And also this cool thing from John's enhanced FIB SEM system is implemented there. So from time to time you little bit overfocus, a little bit underfocus, and if resolution improves, you tune the focus accordingly. Okay, so if you think that FIB SEM Maestro could help you, here's the link to the repository. However, there is nothing yet, so it will be, there is countdown, so it will be hopefully, it will be available for users by end of this year. So meanwhile, I would recommend to add yourself to watch list and be and to stay informed. So as a conclusion, cryoimaging is cool. So you have a low artifact, and it's really streamlined the sample preparation. You can do also the cryotomo together, and the fluorescence preservation is also a feature. Also some drawbacks, low contrast of proteins. You need complex imaging optimization and so on. So with this, I would like to thank all the people included in the project. Also all our customers that gave me permission to show their data. Also thank you for Brno region. And thank you for my boss giving me this strict look that forced me to work better and faster. And thank you for your attention.
Thanks, Pavel, for a fantastic talk. Any questions from the audience? We'll start with Maud.
Thank you very much for your presentation. Can you give a little bit more detail about why changing the working distance during acquisition has been a challenge?
Yeah, like during Cryo FIB SEM, we swing because we have perpendicular imaging. We swing the stage, so it is just movement in one axis, one axis like beta or alpha. What is it? It's just tilt axis. And if you would need to also decrease working distance, it would be like movement of Z and also Y on pre-tilted shuttle, also Y axis. So it will be some complex movement, and I would expect that it will go out of focus most often, most frequently than in just one. Yeah.
Very interesting talk. I have a question on the actual milling source. So you showed different sources you used from gallium ions to different gases. Then I was wondering, have you also looked at the surface topology of your milled section? So do different sources actually affect the local, let's say, etching rate of the surface? I could imagine if you have a slightly higher energy impacting your sample, that you would locally actually allow water molecules to etch. So you, let's say, in classical etching, you remove some water of the surface area, and your sample actually starts sticking out. If you see differences in there, if you look at that, that could also give you more contrast in the way. Also cause some artifacts. Yeah, understand. So I didn't show this data, but we consistently see that with reactive milling, so oxygen and nitrogen, there is a little bit higher contrast than on others. So I'm far from interpretation why, but maybe something with this etching thing. But we measure it also on lipids. That it was like really simple. No, not lipids. These kind of lipids. That was quite good model for that, and we saw this trend as well. So, but it is like negligible. It's not big difference. For example, in the resin sample, the difference between oxygen and xenon is huge, right? It's like this oxygen and reactive and nitrogen really improve the surface quality dramatically, but in case of ice, it's relatively comparable.
Any more questions? Yes, maybe I can ask a quick one. So I was really fascinated. You showed lots of different biological specimens. How much work have you found it, in this setting where you're working with lots of different specimens to optimize samples in terms of the sample preparation, going back to this morning session, and then in terms of the imaging?
Yeah, so in terms of imaging properties, all these data you saw are on more or less the same conditions. So 1 kV and 25 or 13 picoamps, some high line integration, low delta. And I believe, for example, for brain, if you would play around a little bit, you could probably optimize it a bit better. And regarding to sample preparation, yeah, you have to choose right cryoprotectant, and that's it.
That's great. Thank you so much. And we can one super quick question.
For the high pressure freezing, do you have any form of quality control that if you put your sample in the SEM, you can see before you actually start milling, this is okay, or this is just utter crap?
You don't see like light devitrification. You can see if it is like totally not freeze, like without any structure preserved. But for verification of vitrification, you need to go to TM. Like, we saw a lot of nice images in SEM, and if we went to the TM from the same place, we saw some devitrifications. Maybe it happened during transfer, but for the final devitrification verification, we used TM.
A great topic probably for the discussion session later. So thanks again, Pavel, for a great talk.
Thank you.